Good. So let's start here. So talking about CERO is extremely special instrument, which we launched 2019, and we have quite good customer feedback on it. Skip this. Talking about working in 3D, there are a lot of lack of standardization. For many people, it's quite expensive. The handling is complicated. Yields are low. People are not happy with viability. Sometimes the maturation is not good, or they don't find the polarity they would expect from natural environment, and so on and so on. And there, the CERO 3D is a real game changer, as you will hopefully see in a couple of minutes. So this is the instrument. It has a touchpad display. It's rather compact. That means we have users that just put one or two on top of an incubator and use it there. This picture gives you an overview. So within the instrument, it's a full incubator, standalone. So we control temperature, we control CO2, and we also monitor pH, which is not shown in this scheme. So we have an optical control based on phenol red. The tubes as such are rotating bidirectional. And yesterday I had a nice example. It's like that keeps the big structures, especially good for stem cells, for iPSC expansion, ESC expansion, MSC expansion. Primary cells works as well, but there is biggest added value, or organoids. It keeps them free floating. And at the same time, it supplies perfectly with O2 and nutrition, and metabolic stuff is washed away. So they're free floating, and it seems to be for the cells the perfect situation. And it works, and it's easy to use. Now you see here a bit better. We have the fins when it rotates. The cells or organoids are floating. The handling is so easy. You just take the tube out, go to the bench. During that time, the cells, the organoids sediment. You aspirate, you add fresh media, and then after about 4 minutes per tube, you put it back there, continue rotation, and that's it. This is a 50 ml tube, and you can have up to 5,000 organoids in one tube. So the yield is really huge. So it's amazing. But yield is not everything, and handling is also not anything. I'm not sure how much people are here from biotech. They may be interested, especially in the standardization and less of handling time. What everybody should be interested in is in the scientific results. And I choose here an example which was done in the Max Planck Institute in Germany. They are looking at the differentiation of, in this case, is mouse embryonic stem cells into cardiomyocytes. So what you see here is a comparison which they did between CERO 3D and orbital shaker. Now, and day 0, they started the differentiation of the stem cells into cardiomyocyte, and what you see here is on day 3. On the case 0, we have a quite nice homogeneous distribution of the starting aggregates, while here it's a bit more heterogeneous, and if you look precisely, you see also a lot of grain, which are single cells, which have not been included, and so on. So there's already a difference. That is even stronger if you compare after day 8, where the cell population in CERO has been growing dramatically. The density has increased, but still you have a high level of homogeneity. Just keep in mind, we give the stem cells into a tube, and they form this aggregate automatically. So there's nothing you have to do on top. There is an example of what you can do, but you don't have to. And there's a lot more heterogeneity here on the orbital shaker. What is even more important, on day 8, they start the selection cardiomyocyte adding neomycin. So cardiomyocytes will survive only. And what you see here is that basically in this couple of days, the cells in the zero have differentiated to cardiomyocytes, so they stay there. It's a nice high population even after day 13, while the population in the orbital shaker goes down dramatically. So the efficiency, it's not only that the growth of the cells is limited, also those have not differentiated to cardiomyocytes. So which is a big problem for people working on heart diseases. That not only is true for comparison between the orbital shaker and zero, this is also published data from a group which has, I think, 20 zeros. They are using standardized to even commercially produce them. By the way, at this point affordable, there's a post on their website where they say that they save about 70%, 70% of their consumable costs like pipettes, plastic stuff, including media, matrigel, using the zero. But that's just on the side information. Here it was more, this is taken from this paper. They were looking at the expression of specific cardiomyocyte marker or cardiac markers on day 7 and comparing hanging drop, so classical methods, versus zero. And I mean, I think it's not necessary for me to explain what you see here, just at the expression level, like we saw that already on the orbital shaker, it's supported by this results, that the results, the differentiation is much, much better in the zero versus hanging drop or versus orbital shaker. Plus, you save money. And it makes it easier and you use less handling. But another thing is obviously not only the cell volume, but you also want to see something like the vitality. And for the vitality, I'm back on the mouse embryonic stem cells. I will just make a small GIF comparing the contractionality of the cells. And as you can see easily, those grown and differentiated in the zero, they're all active. They're all contractional. They're all doing something. So it's quite high vitality compared to down here, where you only have a little bit here, a little bit here. So when I was looking for the section of the bigger picture, I was already looking for those areas where there was at least a little bit going on. And this was done, as said, by the group of Professor Brown at Max Planck Institute, so also a very, for heart and lung disease, it's probably one of the best institutions, at least in Germany, for this kind of research. The whole thing about using organoids or spheroids or even working with tissue is to mimic in vivo situations as good as possible. And this is another example. So before we had the one with the hanging drop was human iPSCs. The one before from Max Planck was mouse ESCs. This is now an experiment or a process where the group from Technical University in Munich used adult stem cells. By the way, just because, yeah, it is so. Technical University in Munich is the best university in Germany and I think number 50 globally. So it's a very highly recommended university, and they use the zero for infection of Helicobacter pylori. What I also should say is, and that caused some attention of me this year, all these groups bought the first zero or the first 2 zeros last year. And this year they added 1, 2, 3, 4 more instruments. So it really works for them, and they're investing in this platform to expand even their approach. But going back to this result, what is important for them, they are looking at this Helicobacter infections, and for that they need gastric organoids. And gastric organoids usually have this very nice single layer of cells, heavily folded. And you can see in this staining quite well that it works. And we also got from them a comparison where they were looking for the markers for stomach, basically, and they compared colon markers, stomach markers, and you can clearly see that this is really stomach and not colon. So they are quite happy with the system. And they're also, I think from them, the statement was that they save 50% of their cost. Also significant. Finally, we have this guy. Actually, this was the one from the Casey presentation we talked about yesterday. He got the first Casey this year. He bought another two. He is probably one of the highest ranking publishing people globally. So he has, I think, this year 6 or 7 papers, 3 full Nature Review, and some other stuff. And he, for example, discovered the impact of liver cancer, um, the reason of liver cancer, whether it's fatty driven or not fatty driven, in response of immune therapy. If immune therapy is an improvement to treatment of cancer, or like if it's a liver-derived cancer, that it even harms the liver more if you go on immune therapy. Very nice, interesting paper. Anyway, going back here, so they're using the CERO quite heavily, and this example was, and they have 2 approaches. One is more the standard cancer, but they also have the field where they look at virus-induced infectious and cancer. And in this there, they had they're forming or were forming spheroids from liver cells. And the problem there is normally after, if you have spheroids from cells and you keep them in static culture, they go down. So after 15, 20 days, you find necrosis, which is a problem because you need to mature the cells before you can start and use them in bioexpression studies. These data here he shared were made from incubation in CERO after more than 80 days, which means 3 or 4 times longer than normally cells or spheroids will survive in other technologies. So for them, it's really a big game changer. I mean, this is a statement from him, and you see proliferation is going on, still proliferating. There's no indication of any apoptosis, and very important for him as well. All cells are albumin positive, which is a clear indication that all have differentiated, which is important for them as well. So for him, it's really enabling him to do things which he could not do before. And this is just some things. So they have this homogeneous organoids and spheroids in this case, and then they add hepatitis C virus, and then they track how the infection spreads out in the spheroids and under different conditions or treatments. Sorry. Good. To get sometimes what they do is they don't go giving the cells directly to the CERO so that they form the aggregates automatically. For this study, he was even preforming. Oh, sorry. He was even preforming the aggregates. And what you can use there is low attachment plates. So low attachment plates, you give 1,000 or 5,000 cells per well. They are U-shaped, so let them sit there for a couple of days, 2, 3, 4, 5, depends on the cell. And then you transfer them to CERO, because then you have extremely homogeneous starting spheroids, which continue to be extremely homogeneous throughout the process. That means your results even get more meaningful. For my colleagues in Malaysia, we just have them new in the portfolio as well. So what we have here is it's a new coating which has been developed by one of the biggest chemical companies, which is sitting not far away. I'm close to Heidelberg, so it's not far away from here. And actually it was for some technical approach, but then they found out that it's working extremely well with cells. And also adds a lot of added value to life science experiments. This special coating for low attachment plates. Currently we have 96, 384 is close to come. We have now a lot of requests for 24 well, so we also will look into this quickly. Now, question, what have we done? So there's already a lot of experiments and some pictures here. So based on stem cells, what is extremely good is the expansion of MESCs and iPSCs. It's so easy that we now also will go into a cooperation with guys in Japan, in Kyoto. Not the CERO, but the foundation that they founded for production of iPSCs. So majorly iPSCs and iPSCs from mouse and humans. And what you see here is basically the aggregate, the spheroid of ESCs or iPSCs, so different kind of illumination. And as you see, they're already in very nice homogeneous aggregates, embryonic bodies, some people call them. So you can, after expansion and after generating a lot of cells, you can directly take them in differentiation. So you don't need to form the bodies itself. The spheroids are already a result of expansion. So it's very simple. Now, question: any idea why all these are majorly in mouse and human, and MSCs are majorly taken from bovine and swine? Come on, any idea? Okay. These are primarily studies which are going on in many countries for artificial meat. And MSCs play a big role in artificial meat. And people are looking to generate high amount of MSCs in zero, but obviously then from those species where you want to have your meat from. So another example here is primary cells. I don't want to run to, you can read yourself. So quite broad, different kind of cells. And we have organoids from cardiac, from liver, from brain, from lung, from intestine, from stomach, from retina, from different species. So you see here, retinal, a lung one. You remember the intestinal? These are early stage organoids before they have differentiated and show this high folding. Now this is a nice one, because I didn't understand the results, and I said like, yeah, nice staining, but not sure what this immunostaining means. Until I was we had a bit of contact with this user of the CERO, and I don't know who is from you working in cancer, but this is a lab which is close to one of the bigger hospitals, and they get tumor tissue from patients. And the problem that he told me is in what they're using is for the optimal strategy of treatment. However, when they take out the tumor and cultivate it in vitro, to make it in simple words, the tumor starts to forget it's a tumor. So the expression levels of tumor markers goes down. And now using the CERO to maintain the tumor tissues, what they said, the tumor does not forget it's a tumor, so it maintains all this expression, which makes it more likely that the therapy efforts or the results of the therapy testing is much more meaningful than with other methods. Good. So just some examples for tissue. This is rat liver, here mouse liver. Finally, slides. This is expansion of iPSC. This is also proven from CERO. They do the same. This is published here from Bonn. You digest, you put them into the CERO, and then in 5 days you can have an expansion of 22-fold. Rather use a very little amount of media, no matrigel. Most of the things are not require any matrigel. They maintain pluripotency, which I've not shown here, but it's in the paper. But they can still differentiate in all 3 germ layers. So that was a quick overview about the CERO. Questions?
Any questions for Dr.? Please do ask if you have any questions for Dr. Joachim.
Okay. Anyway, you can follow. I mean, you can talk with my partners in Malaysia anytime, and I think we will be happy to answer all your questions. And also, maybe if you share what you're doing, we can get even more dedicated information for you.
Sure, for sure, we will help. Thank you, Dr. Joachim.
Thank you very much.
It's really a comprehensive presentation for you. Thank you for
Thank you
joining
very much.
today.
And please feel free to contact us and with any questions you have later on.
Sure, Dr. Joachim.
Okay, bye.
Thank you. See you again. Have a nice day.